To speak. I love this meeting. It's really so informative to the scientists and to the families, and it's a win-win for all of us. So I am a pediatric oncologist. I trained at the Children's Hospital in Philadelphia, CHOP, and then I moved to Boston many years ago, and I've been at Dana-Farber Cancer Institute for 30 years. And I've been studying for 30 years a rare genetic disease. Genetic disease is called Fanconi anemia. It's a pathway of genes, as I'll show you in a moment. And I think that this pathway, which I've been studying, which regulates DNA repair, is highly relevant to the study of the CHAMP1 pathway, which we've been hearing about today. So with that introduction, this is the gentleman in my laboratory who's done pretty much all of the work I'm going to describe right now. His name is Feng Li. He's in China right now, and I certainly hope he gets his visa so he can come back to my laboratory. He's a really wonderful investigator. So this is the Fanconi anemia pathway. And just to give you some idea, I have invested my career in this pathway for the last 30 years. And what's clear in this pathway is there's 23 genes. Each gene encodes a protein, and these proteins work together in a pathway, and this pathway regulates a process called DNA repair. So if a child is born who knocks out any one of those genes, you get the same physical appearance of these patients, the same anemia, the same cancer predisposition, and the same DNA repair defect. So knocking out any one of these genes gives the same phenotype. Similar to the knockout of POGZ and CHAMP, which gives a very similar physical appearance and molecular appearance. So one of the genes in this pathway is a gene called Rev7. It's down here in the bottom of the pathway. And this gene, it's also called FANC-V, Fanconi anemia type V. This protein has always been particularly interesting to me. It's a small protein. It's only 20 kilodaltons in size, but it's an abundant protein. And we have found over the years that every protein that Rev7 binds to is involved in a DNA repair process, okay? And indeed, that was the case. So what Fang did some years ago is he took the Rev7 protein, this Fanconi protein, and he pulled it down from a cell extract. And when he pulled it down, not surprisingly, the protein bound to some known DNA repair proteins like SHIELD3, it binds to Rev7 itself, it binds to Rev3. But the interesting thing was it bound to these two new proteins, which I didn't know anything about, CHAMP1 and POGZ, okay? But I surmised that this CHAMP1 and POGZ probably had something to do with DNA repair. So that was the hypothesis. So indeed, this is a NMR structure of the Rev7 protein. And you can see here the Rev7 protein binds to Rev3. That's involved in a DNA repair process. The Rev7 protein binds to SHIELD3. That's involved in a DNA repair process. And on the far right here, yes, Rev7 binds to the CHAMP1 protein. And that's involved in a very specific kind of DNA repair called homologous recombination. Joining the group here. And we showed this some years ago now. And in fact, what we showed is if you take cells now, CHAMP1 promotes this process of homologous recombination. How do we measure homologous recombination? By an assay, which some of you know, called Rad51 foci. And that if you knock down CHAMP1 protein, you reduce the level of Rad51 foci. So that's really an evidence that CHAMP1 protein really was required for normal homologous recombination. And importantly also that when cells have a defect in homologous recombination, they're sensitive to a drug called olaparib, which is a PARP inhibitor drug. Okay, then that comes back later in my talk. All right, so how did we show this? We showed this, again, Fang Li showed this a few years ago now, back in 2022, in a Cell Reports paper, that when he immunoprecipitated the protein SHIELD3 and then blotted for Rev7, yes, indeed, SHIELD3 binds to Rev7, but when he knocks out the CHAMP protein, knocks it out, gets rid of this protein, now you get an increase in the expression, the increase in the co-precipitation of the SHIELD3 protein and the Rev7 protein. And that allowed us to conclude that the way the CHAMP1 protein is working, okay, is by stealing Rev7, actually stealing the Rev7 protein from the shieldin complex. So this is a molecular function of the CHAMP protein. Okay, so that said, Okay, but then, okay, so we had this feeling then that that CHAMP protein was involved in homologous recombination DNA repair. An exciting finding and a brand new finding, and for me, a brand new DNA repair syndrome that I could work on in my laboratory. Okay, but then we went back to this paper, and you've seen it on a few other slides this morning, that published in Cell back in 2010. It turns out that the CHAMP1 protein and the POGZ protein co-precipitate, and they're very closely affiliated with each other with another protein called HP1 alpha. This is a protein complex which is found in heterochromatin, the tightly packed chromatin that Irini spoke about earlier this morning. Okay, now, so we wanted to sort of study this CHAMP1 complex more closely. And all the stuff I'm going to show right now is now posted in a paper. We recently posted this paper in a journal called BioArchives. The paper's now under review elsewhere, but it's posted on BioArchives. And what we showed in this paper is that, in fact, the CHAMP1 protein, shown here on the left, is bound to POGZ. And there's a very interesting interaction between the two proteins. The N-terminus, or the left-hand side of CHAMP1 protein, is bound to the carboxy terminus of the POGZ protein, okay? And all of these proteins interact in a big complex. And we know a lot about this complex now because this complex is, it's been proven by co-precipitation of proteins using mutant proteins, but it's also been proven by using AlphaFold, which is a informatics way, a computational way of looking at protein-protein interaction. All right, so this raises the very interesting question. Okay, here are these proteins interacting in a complex, CHAMP1, POGZ, HP1 alpha, maybe other proteins that we heard about this morning, like ADNP or ARID1B. Maybe these proteins are all part of a complex, but what is this complex actually doing? Okay, so it turns out, and now this is I'm going to give you the answer first. The answer first and tell you sort of how we figured it out, how Fang figured it out, is the following. We think that this complex, which is a heterochromatin complex, binds to centromeres and binds to telomeric regions of chromosomes, that this complex is a machine. It's a non-enzymatic machine. It's a structural machine which binds to an enzyme called CETDB1. CETDB1 is a histone methyltransferase. And this machine, POGZ CHAMP1 machine, recruits the CETDB1 enzyme to heterochromatin. Okay, there the complex does three things. It increases heterochromatin assembly by causing an increase in H3K9 methylation locally. Now that process, number one, actually causes compaction of the chromatin. So you might think that that would decrease DNA repair and decrease transcription, okay? But in fact, it leads to an opening of DNA repair. Because now that the heterochromatin is compacted, number two occurs, and the heterochromatin, the telomeres, cluster together at one site in the nucleus. And that clustering also is mediated by, guess what, the CHAMP1 complex. And then finally, once that clustering occurs, now homologous recombination DNA repair can occur. So the CHAMP1 complex contributes to all three of these events. Okay, let me show you some of that. Okay, so for instance, here's an example now of looking at the methylation of H3K9, which is a enzymatic reaction which results in the assembly of heterochromatin. And indeed, what you can see here is, in response to DNA damage, heterochromatin is assembled, and you get an increase in H3K9 methylation. But if you knock out CHAMP, or if you knock out POGZ, you get a decrease in this heterochromatin assembly, and you get a decrease in H3K9 methylation. All right, so that was interesting. Now, the second thing was this cluster formation. And to study cluster formation, we use an enzyme which is FOC1. It's an enzyme which cuts double-strand breaks in DNA. And we can deliver this DNA cutting enzyme specifically to telomeric regions. And when we do that, we actually find that in response to this double-strand break, now telomeres are clustered together. Telomeres from multiple chromosomes are clustered together into one site. And this clustering requires the CHAMP complex. So if you knock out the CHAMP complex, you cannot cluster these telomeres in one site. And then finally, number 3, the CHAMP1 complex is required for homologous recombination repair. As Kozo has shown in the past, as Alex has shown in the past, this complex is required for homologous recombination. And if you knock this complex out, you get a decrease in Rad51 foci and a decrease in homologous recombination. Okay, so, and how does it do that? Rather than get into the details, it turns out that the CHAMP1 complex at a telomeric site, at a heterochromatin site, it ends up recruiting a lot of homologous recombination proteins to that site, including BRCA1 and BRCA2 and Rad51 are recruited to that site, and that's how this CHAMP complex is promoting homologous recombinations at this heterochromatic site. Okay, so I told you then that, just review what I just said, that the CHAMP complex does 3 things. It increases heterochromatin assembly and H3K9 methylation, it increases telomeric clustering, and it increases homologous recombination repair. Well, that's interesting because as a cancer biologist, I'm very interested in a pathway known as the ALT pathway of telomere maintenance. It's well known that some cancers lengthen their telomeres by using this alternative telomere maintenance program, and that program requires heterochromatin assembly, it requires telomere clustering, and it requires homologous recombination. And essentially what we've now gone on to show is this CHAMP pathway, this CHAMP protein complex, is required for the maintenance of this alternative pathway for telomere elongation. And that's important because what we've done now, I think, is we've connected the mainstay of CHAMP POGZ biology to cancer biology. In my experience, if you can bring those 2 fields together, you will increase the number of scientific investigators who work on CHAMP1. And that's a good thing for us. That's a good thing for the field. It's a good thing for fundraising, and it's a general good thing for the field. Okay. So that said, we can skip that and just simply say, this is interesting that if you knock out the CHAMP complex, these telomeres shorten very rapidly. So in tumor cells, in cancer cells that use the alternative mechanism for telomere maintenance, now if you inhibit or block the CHAMP pathway or inhibit CETDB1, which is the methyltransferase, these telomeres rapidly shorten. So in fact, this is a potential treatment for cancers that have that use the alternative mechanism of telomere maintenance. All right, so again, this is the cluster formation I was telling you about. This CHAMP1 complex now recruits this protein CETDB1, which is an enzyme. It brings this enzyme to these telomere clusters. And now this enzyme could increase the level of expression of this H3K9 methylation. So the pathway is using these proteins as a tool for bringing the methyltransferase to the heterochromatin. All right? And you can see that, in fact, if you knock out CHAMP with a guide RNA using CRISPR, or knock out POGZ on the far right, now there's a significant reduction in the amount of CETDB1 protein which is recruited to heterochromatin. Okay. Now, and then we could use a new tool called AlphaFold 3 in this case, where we can really show that the CETDB1 protein binds very tightly to the amino terminus of POGZ. So POGZ really seems to be a critical recruitment factor that's binding to this methyltransferase and targeting this methyltransferase to sites of heterochromatin. Okay. All right, so this in the last 3 slides, a little bit about the cell lines that Kozo just described. And just to give you some idea, these cell lines have been incredibly valuable to investigators in the field. So these cell lines were established with blood samples that were taken from children with the CHAMP syndrome over 8 or 10 years ago they were taken. These cell lines were immortalized, and these cell lines are now freely available to academic investigators who work on the CHAMP syndrome. And you can see that these cells, just as Kozo just showed, these cells derive directly from patients with CHAMP syndrome have expressed these truncated CHAMP proteins. They have expressed the full-length CHAMP protein and the truncated CHAMP protein. And again, just as Kozo just said, and I'd like to see our data validating each other. I think he likes to see that too. You can see here that these blood cells now taken from children with CHAMP syndrome have a decrease in H3K9 heterochromatin assembly, and they have a decrease in homologous recombination, which is easily measured, and an increase in olaparib sensitivity, which is a drug which can be used to assay the blood. So the point here is the way up till now, the way children have been diagnosed with CHAMP syndrome or White-Sutton syndrome have been diagnosed based on the basis of a mutation in either the CHAMP1 gene or in the POGZ gene. Now we can actually take a blood test and do a blood test looking for DNA damage in the peripheral blood and make in a way, a more rapid diagnosis of whether or not the child has the CHAMP syndrome or the WSS syndrome. And we're going to start using that blood test as a correlate to the genetic sequencing, which of course is now routinely done for all children with autism. Okay, so in conclusion then, important summary slide. This is what I said, and I know this is dense science. This is a basic science talk. I give the same slides to a talk, with DNA repair experts. Now I'm giving it to an audience with a number of lay people and parents, but there's a very important, and I can't simplify it. You can only simplify this stuff so much, okay? But these are the important points of my talk. That the CHAMP complex promotes this methylation, this enzymatic change in a histone protein called H3. And this results in the compaction of chromatin, heterochromatin assembly. Second, the CHAMP complex promotes telomere clustering. It brings chromosome ends into one site. And when it brings these chromosomes ends to one site, in a way, this enhances homologous recombination and enhances transcription. Okay, this is how clustering of a heterochromatin can actually enhance processes that were previously thought to require the unfolding of the chromatin. Third, that the CHAMP1 complex promotes this DNA repair process. Fourth, that the CHAMP complex recruits CETDB1 methyltransferase to telomeres. Lastly, this CHAMP complex is required for the ALT pathway, which brings our study of this disease closer to the understanding of cancer. And finally, that lymphocytes, blood samples taken from children, young adults with CHAMP syndrome or White-Sutton syndrome, will be incredibly useful to investigators in this field. So I strongly urge families to work with Irini and others who are trying to collect these cell lines for the use of a lot of investigators. And that was a lot of foresight on the process of the early people involved in the CHAMP syndrome, Jeff and Amanda and others who have been able to collect those cells over time and make them freely available to investigators in the field. So I'm very grateful for that. These are investigators who did the field, but most notably Fang Li, Amira El-Bakri, and Aleem Saeed did the work that I just showed. So I'll stop here and answer a question. Thank you.
Before taking any question, yes, we are trying to solve the noise problem. Seems that asking decently to lower down the volume was resulting in higher the volume. So you can understand that it is an awful situation. Sorry about that.
Despite the noise, beautiful data and fantastic talk.
Thank you.
So I mean, and I think we see the same data as well in our case that POGZ and CHAMP1, rather than just stabilizing methylation, is actually participating in a process, it's a dynamic process. And actually when we tether POGZ to the LACO array, we also do see an increase in H3K9 trimethylation, which goes along with what you're saying.
Nice.
So I guess I have two questions.
Is that published?
No. It's actually part of my grant application, so it's not published.
Good.
So, but we do see the same thing.
Great. That's happy to hear that.
Totally making sense about what you're seeing. So I guess I have two questions. The first question is that, do you see other trimethylation changing at, so for example, H3K27 trimethylation, does it also change when you actually affect CHAMP1 set BP1 interaction? And the second one is, CBP1 is actually, if I understand properly, only adding the third methylation on H3K9. What about the mono and the di? Is there also an active process of CHAMP1 in allowing the initiation of methylation in the process?
Yeah, so the mono and the di might already be there.
You think so?
You know,
Okay.
so I'm, you know, and I think that, and we have not looked at H3K27.
Okay.
We've not looked at other histone modifications. We specifically focused on H3K9 because we knew that it was a heterochromatic process, but I think it would be good to go back and do that.
Because, so when we did the bio-ID of POXZ, we get only the EHMT1 and EHMT2, which are the mono and the dimethylated enzymes. So which we don't get.
Right, but it sounds like your work is very consistent with the idea that POGZ is a recruitment factor that's bringing the methyltransferase to the site.
Agreed.
I'm happy to hear that. Yeah. Good.
Hey everybody, before we get ready to questions, I just want to take a quick pause and kind of address the noise. I want to say thank you, Dr. Tanaka and Dr. Deandra, for your poise. I don't think you could possibly handle the noise better as an amazing researchers as you guys are, and just your composure as human beings. So thank you so much, and thank you everybody for your patience. I just wanted to, we'll continue with the Q&A. For the next speaker, if we want to take a very brief intermission while we just try to resolve the problem and see if there's a solution, because it doesn't seem to be going away, we might take a brief intermission, and I'll leave that up to the next speaker, and maybe you guys could give your opinion on that. Thank you. I just wanted to make sure.
Oh, yeah.
Really appreciate you so much.
Alan, great talk. Thank you so much. And I know it's becoming a recurring theme, but I just wanted to sort of present it to the floor and see what you thought. If I could make you speculate a little bit, I mean, obviously, as everybody keeps bringing up, neurons don't have homologous recombination available to them, right? But I'm sort of curious if, you know, via this heterochromatin packaging that you're seeing, do you think that there's any chance that you could also increase the amount of homology that's maybe present for, say, another repair mechanism like non-homologous end joining that neurons might be using during their repair mechanisms?
Yeah. No, I like that idea, and I think neurons do have an error-free DNA repair mechanism that we just don't understand yet. I mean, neurons, you know, if you have big genes that have to be transcribed, if there were mutations in those genes, you would not get that protein expression. So we really need good error-free DNA repair in post-mitotic neurons. We just don't know the mechanism yet. And I think it is possible that this clustering that the CHAMP pathway seems to be providing is contributing to some error-free DNA repair pathway. You know, so I think that's a long-range question, but thank you.
Yeah, thank you.
Hi, thank you for the talk. So this might have been covered in either yours or the other presentation about DNA repair, and maybe cell repair, and maybe I just didn't get it, but would we infer that there could be higher cancer incidence rates in CHAMP1 and White Sutton patients?
I think so. I think it'd be a very, very low risk. I mean, I think there have been a couple of cases of children with CHAMP syndrome that have leukemia, for instance, but I don't think it's a super high risk. But I also think there have been some cases of CHAMP children who actually have a decrease in blood cell count, and that could also be consistent with a decrease in homologous recombination repair. So I think the DNA repair abnormality in the CHAMP syndrome is subtle. I don't think it's like a profound DNA repair defect like we see in Fanconi anemia, this other disease I study. I think it's subtle, and maybe the defect is more a DNA repair defect in the brain. You know, but I think that I don't think people should go home worried that their child with CHAMP or WSS syndrome are going to get cancer. I don't think that should be a worry, but it's certainly something that we'll keep an eye on.
Okay, and then one more. So you laid out nicely, like, you know, all these functions that the CHAMP1 complex does, like a series of different things it does. Is there, like, just generally potential in the future for some kind of therapy that could, you know, rather than upregulating the expression of the gene, like do something at the later step in this process, like mimic a functionality, like recruit more set DV1 proteins or something like that?
Right. Reducing DNA damaging agents, ultraviolet light, the dietary agents, aldehydes in the diet, reducing certain things in the diet that might reduce DNA damage would be one way. We could look more at that. But upregulating DNA repair pathway that's otherwise deficient, it would be hard to do. It would be very hard to do. Metformin came up earlier today. That's what we're giving to children with Fanconi anemia for the same reason that we talked about this morning. So, or trying to upregulate the expression of some proteins which are lost in CHAMP1 neurons, you know, indirectly, yeah, through drug modification. Yeah, is that?
Yeah, great talk. I have a question about the clustering of the telomeres. You said that you think that this promotes like, what is the purpose of them coming together? You think it's because it promotes homologous recombination, it boosts transcription? What about the idea that maybe this complex is kind of limiting in amount and there's only really enough to make one spot in the nucleus or a few spots where this could be possible? This might explain why haploinsufficiency, not having enough to be able to make one of these spots happen.
Yeah.
Do you think?
It'd be interesting to know if haploinsufficient cells have a decrease in those clusters, and that would be testable using your heterozygote cells. So I think that's plausible. But to me, it was counterintuitive. It was that you'd think that in increasing the compaction, the packing of these heterochromatin regions would shut off transcription and shut off homologous recombination repair. But it allows this clustering to occur, which then can open up the process, I guess. So that's kind of the, yeah.
Hi, Alan. Hello.
Oh,
yeah.
Yeah.
So in your model, so the role of this CHAMP1 POXY complex, so at least the heterochromatin region for homologous recombination is not direct, but rather indirect. So that by so the clustering the substrate, so the cross together. Is that right?
I think that's right.
Okay. Do you have any?
The clustering is bringing DNA sequences next to each other to allow recombination to occur.
Do you have some direct evidence that these complex cross-link different DNA strand? So that not only cis, but it's in trans, so that cross-link different DNA strand together. Do you have some direct evidence?
No.
Okay.
No, we don't have direct evidence for that.
Yeah, thank you very much.
Okay. Oh, there's one in the back. A telomere question from the back.
Hi, sorry to be so predictable. Do CHAMP1 kids have shorter telomeres? Do CHAMP1 kids have shorter telomeres?
That's a good question. No, we're looking at that right now. Yeah, no, we're testing that right now.
Any idea so I can sleep tonight?
Yeah, that would be consistent with certainly what we're seeing here. Yeah. But the telomere shortening occurs especially in these tumor cells that are using this alternative mechanism of telomere maintenance. I'm not sure that if a telomere is lengthened by telomerase, I don't think that the loss of the CHAMP pathway would reduce the size of the telomere. But it's a good question. We'll take a look. Yeah.
We have a remote question. I'm just going to read it out. Significance of this for a child with POXY and BRCA2. Is there any significance between, or to what your talk and POXY and BRCA2 together? I'm assuming BRCA2 is a gene.
Yeah, no, BRCA2 is a gene that's involved in homologous recombination. I don't know of any situations where those two genes are both knocked out, so I don't think there's any obvious crosstalk between those two genes. So.
Okay, thank you.
Good.